So hi everyone and welcome. I thought it might be nice to add to TJ's background of my scientific career is that I have only started using remote sensing in my science about 4 years ago. So I was a novice at some point. You have to start somewhere learning, but you can get quite far if you dedicate time to it. So hopefully my lecture will help you with that. And so today I will be talking about the use of multispectral remote sensing observations to monitor water quality and to map floods. And the work that I'm going to present to you today is not just my own, but there are many other scientists involved, and it's very much a team effort. Before I dive into the examples from our work and research, I wanted to start off with repeating what exactly Earth observation and satellite remote sensing is. You have had this in the previous lecture, but I just wanted to remind you in a couple of slides to set the scene for the work I'm going to present. So, remote sensing is the term we use for gathering information about an object or a phenomenon without making physical contact. And Earth observation is gathering information specifically about the Earth's physical, chemical, and biological systems via remote sensing technologies. So, Earth observation can be carried out using various platforms, including satellites, planes, and drones. And I will be mostly talking about satellite observations today. So as you have already learned in module 1 of this training course, there are many satellites in orbit, and around 800 of those satellites are Earth observation missions for weather, land, marine, and cryosphere applications. So satellites will either have active or passive sensors, and the active sensors transmit their own energy towards the Earth and measure the returning signal, while the passive sensors measure radiation reflected from the Earth's surface, such as visible and infrared light. An important distinction is that the active sensors can penetrate clouds, while the passive sensors cannot see through clouds. And I will come back to this later in my presentation when we compare these two types of sensors for mapping of floods. The satellites I mostly use in my research have passive sensors that measure the color of the ocean and other water bodies. As Lauren and Anna explained before, the color of the ocean results from the interaction of incident light with substances in the water. And light can be absorbed, transmitted, scattered, or reflected. Satellites that observe ocean color were originally designed to map distributions of phytoplankton in the ocean. But we now have many more applications of these satellites. And the information contained in ocean color does not only include the concentration of phytoplankton, but also other so-called optically active constituents. So in this image, which I think has been shown on this course before, we see chlorophyll, which is an indicator of phytoplankton biomass. So we see water, color dissolved organic matter, and sediments. And all these substances in the water have different colors that we can observe. And I just wanted to share the image of a coccolitho bloom near Plymouth, where I live, because it's such beautiful, and it also shows that phytoplankton doesn't always occur as green, but can have quite a bright blue-greenish color as well. So ocean color satellites observe the reflected solar radiation from these different substances in the water. And each of these substances in the water have a distinct spectral signature. Confusingly, we speak of both ocean color and multispectral remote sensing when referring to this technology, with the ocean color satellites specifically designed to observe the marine environment and the multispectral satellites designed to observe the terrestrial environment. These multispectral satellites are very useful for high resolution observations in coastal areas, which has already been mentioned a couple of times in this course. So we use these satellites to study aquatic systems as well. The clear benefit of satellite remote sensing in environmental studies is the scale. There's high spatial and temporal coverage, and long time surveys are often available. And as Lauren mentioned before, the latest ocean color satellites have a spatial resolution of about 300 meters and observe the global oceans. and the latest multispectral satellites have spatial resolutions of 10 meters, but the revisit time is 5 to 12 days, depending on where you are. There are also many commercial satellites available that will have finer resolution, but of course these are paid for services. And so both ocean color and multispectral remote sensing satellites have been around for decades, with the first satellites launched in the 1970s. In the case studies on water quality and flood mapping that I will be presenting in the rest of this presentation, I will be talking a lot about 4 satellite sensors: Landsat 8, Sentinel-2A and Sentinel-2B, and Sentinel-1A. The Landsat and Sentinel-2 sensors are multispectral imagers, and they are passive sensors that are based on the technology I just discussed. So there are limitations in these sensors, and importantly, we cannot see through clouds. We also use Sentinel-1 sensors, which is an active sensor using synthetic aperture radar, or SAR technology. And this satellite does not see the Earth as we do, or multispectral sensors do, but instead the signal is responsive to the surface characteristics like buildings, vegetation, and moisture. And so one of the things SAR is used for is the mapping of water, which is obviously really important when you study floods. Before I dive into the case studies, I wanted to talk about our study region. So the region that we carry out our research is the Vembanad Col wetland system in Kerala in India. And Lake Vembanad is a large lake of approximately 100 kilometers long on the southwest coast of India. The lake and the surrounding wetland area are known for its natural beauty and are protected under national and international regulations, including the Ramsar Convention. The lake forms a really important resource for local communities and is used for coastal and freshwater aquaculture, capture of fisheries, Vocali rice cultivation, tourism, among many other activities. But Lake Vembanad is also under pressure from all of these anthropogenic activities, and that leads to domestic waste and sewage discharge, industrial pollution, including heavy metal contaminations and eutrophication, which all lead to poor water quality and aquatic weed infestation. In the past 5 years, a team of scientists from the UK and India, including myself, have been studying Lake Vembanad in relation to human health, with a focus on the environmental controls of waterborne diseases, such as cholera, which is endemic in the region. And during those 5 years, we've seen some major events, including the COVID-19 pandemic and severe floods in August 2018, and very heavy floods between 2019 and 2021. And I'm going to discuss the effects of these events on the aquatic system in the Lake Vembanad region in more detail as part of 4 different case studies. So the first case study is focused on water quality monitoring and was part of a project funded by the UK Natural Environmental Research Council and the Department of Science and Technology of the Government of India. And this project was called Revival. And in this case study, we looked at the effects of the COVID-19 lockdown on water quality in Lake Vembanad using a combination of Sentinel-2 and Landsat 8 data and in situ data that was collected as part of field campaigns. The second case study was part of the same research project and is focused on the development of regional satellite retrieval algorithms for chlorophyll A and for total suspended matter. And for this case study, we used Sentinel-2 data and the same in situ data collected in Lake Vembanad. But we also made use of the in situ data that's available from NASA. The third case study was again part of the Revival research project and is focused on the mapping of Vibrio cholera bacteria in the environment. And Shuba already touched upon this in her presentation last week, and I will show more details on the algorithms we developed for Lake Vembanad. And so to map pathogens in the environment, we made use of Sentinel-2 data and the in situ data collected in Lake Vembanad. And the fourth case study of flood mapping is part of a project funded by the European Space Agency that is called WIGEN. In this case study, we studied different techniques to map the once-in-a-lifetime floods in August 2018, using both multispectral observations from Sentinel-2 and the SAR observations from Sentinel-1. And in this case study, we also made use of local information for the validation of the satellite observations. So let's start with the first case study on water quality. So despite considerable efforts to protect vulnerable marine, coastal, and freshwater ecosystems, anthropogenic activities remain one of the main causes of poor water quality in rivers, lakes, and wetland systems worldwide. It has been shown that a combination of industrial and urban pollution, overexploitation of natural resources, and climate change have negative impacts on not only the marine and freshwater organisms, but also the human populations that are dependent on the aquatic environment for their livelihoods. So poor water quality can lead to adverse ecological and socio-economic impacts. Shubha already mentioned the Sustainable Development Goals of the United Nations in her presentation last week. And in relation to access to clean water and sanitation to improve human health, the SDGs also address the need for sustainable development of vulnerable coastal regions, such as the Vembanot Coral Wetland System. So Sustainable Development Goal 14 deals with the conservation and sustainable use of the oceans, seas, and marine resources. And meeting target 14.1, that is by 2025, so that's very soon, prevent and significantly reduce marine pollution of all kinds, in particular from land-based activities, including marine debris and nutrient pollution, will require as a first step that the quality of water in aquatic bodies is assessed in a routine manner and changes therein are recorded over time. And then target point 14.2, which is by 2020, so that's already in the past, sustainably manage and protect marine and coastal ecosystems to avoid significant adverse impacts by strengthening the resilience and take action for their restoration in order to achieve a healthy and predictive ocean. This requires an assessment of the impact of human activities on water quality. However, it's often not straightforward to demonstrate the direct effect of various anthropogenic activities on water quality. And so during 2020, a unique opportunity arose to study water quality in Lake Vembanot under reduced anthropogenic activities. In the response to the global pandemic caused by COVID-19, the government of India imposed a nationwide lockdown on the 25th of March that lasted until the 31st of May in 2020. So during this period, people were required to stay at home, and most services, including transport, education, industry, fish and vegetable markets, and hospitality were suspended, with the exemption of essential services. And the easing of the lockdown regulations after May 2020 occurred in several phases in India, with restrictions to services remaining in place in containment zones where infection rates and the number of COVID-19 cases were high. So we decided to study water quality during this period of reduced anthropogenic activities using a combination of satellite and in situ observations. We used 5 different indicators for water quality that are amenable to multispectral remote sensing. So the first one is chlorophyll A, which is a measure of phytoplankton biomass. Then second one is total suspended matter, the operational defined as all organic and inorganic suspended particles larger than 0.7 micrometers. And then we have the absorption by color dissolved organic matter, which is the light absorbing fraction of organic particles that are smaller than 0.45 micrometers. So that's dissolved organic matter. Then we have turbidity, a measure of water clarity that is related to chlorophyll A, total suspended matter, and the absorption of color dissolved organic matter. And then we have the Forel-Uhle classification of watercolor, which ranges from blue to green to brown waters, with blue waters termed as clear waters low in suspended and dissolved materials. And these water quality indicators were obtained from two different data sources, from remote sensing observations and from Sentinel, from Sentinel-2 and Landsat-8 sensors, and then in situ observations. We analyzed data from 2013 to 2020, with a nationwide lockdown in India occurring from the end of March to the end of May. The remote sensing observations were divided into pre-lockdown, lockdown, and post-lockdown periods, with observations from May to September typically being obstructed by cloud cover due to the monsoon season. We also used the full time series of the remote sensing observations to analyze long-term trends in water quality. And we had the opportunity to collect in situ samples later during lockdown in May 2020, which we then compared to samples we collected earlier in May 2019. I will come back to this figure when I discuss the results, but I will provide a bit more information on the different data sources first. So for the remote sensing observations, we used data from Sentinel-2 and Landsat-8, which were downloaded from the Copernicus data catalog and from the Google Earth Engine data catalog. And I processed this data using Acolyte, which performs an atmospheric correction based on the dark spectrum fitting, and it provides remote sensing reflectances. And this method is based on open and free data and software, which I think is really important. And I think later in this course, you're going to learn how to access the data and how to process data yourself. To estimate these 5 satellite retrieval algorithms, which are listed in the table. So for example, for chlorophyll A, I used the OC2 version 2 algorithm, which is based on the blue and green bands. And these algorithms have been validated for a variety of water types and have been used in Lake Vembanot before, but their use has not been validated for Lake Vembanot. And I will briefly come back to that in a couple of minutes' times. And so just to give you an example of what the data look like, the maps show the application of the algorithms to the processed satellite data, with examples of the 5 water quality indicators. So from left to right, chlorophyll A, total suspended matter, the absorption by color dissolved organic matter, turbidity, and the FU classification of watercolor. As I mentioned earlier, we've also collected in situ data as part of the Revival research project. So field campaigns took place between May 2018 and May 2019, with samples being collected approximately every 20 days at 13 different locations in Lake Vembanot, which are shown in the figure on the right. And just to remind you of the scale, the lake is about 100 kilometers long. So these field campaigns were quite an undertaking, and it took 2 days to sample all of the stations. And so as part of these field campaigns, the 5 water quality indicators were measured, but also many more measurements were performed, including the presence of pathogenic bacteria such as Vibrio cholera. And in May 2020, the Indian project partners had the opportunity to collect in situ data. So we have some additional measurements during the lockdown period. As I mentioned before, we used existing satellite retrieval algorithms in this study. And it's therefore important to assess whether these algorithms perform well in our study region. And normally you would validate the satellite-based estimates with in situ data using a match-up data set, but our efforts were limited by the amount of match-up data points we had. And the spatial temporal matching of the in situ sampling days with satellite overpasses was low, with about 22 data points for Sentinel-2 and 8 data points for Landsat 8. So we consider this too low a number of match-up data points for a validation exercise at the time. But we did compare the monthly mean values of the satellite-derived and in situ water quality indicators, of which the results are shown here. So overall, the seasonality in the water quality indicators looks similar between the different data sources, but we also see some differences between the satellite-derived and the in situ water quality indicators. So for chlorophyll A, we see that higher chlorophyll A concentrations were not captured by the satellite retrieval algorithm. Total suspended matter and turbidity compared relatively well. The absorption by CDOM was overestimated by about 4 times, and the FU classes were lower in the satellite-derived observations compared to the in situ observations. So we see that some of the satellite retrieval algorithms performed differently in Lake Vembanad compared with earlier reports in literature from other regions. And so as part of this work, we also studied other published satellite retrieval algorithms, but these did not yield a better comparison with the in situ observations. We therefore chose to use the selected satellite algorithms, considering that these algorithms have previously been used for Lake Vembanad, and considering that the results are presented as a relative change separately for remote sensing and the in situ data. After completing this work, we followed a modeling approach to validate and develop regional algorithms for 2 of the water quality indicators. And I will come back to this in much more detail in the second case study. So I will now show you the results of the study. So remember that we are looking at the effect of the reduced anthropogenic activities on water quality during the lockdown in Lake Vembanad. So we started with studying the changes in water quality indicators during the lockdown in 2020 by comparing the remote sensing observations in the month preceding the lockdown with those of the first month of lockdown. And the results showed that water quality improved in large areas of Lake Vembanad during April 2020, as evidenced by degrees, which is shown in blue in these maps, in total suspended matter, in the absorption by color dissolved organic matter, and turbidity. And all of these changes led to clearer waters as indicated by the FU classification of water color. I will continue on showing results at the locations of the 13 in situ sampling stations, which are shown here in the map on the right. We continue comparing the observed changes in water quality indicators during the lockdown period in 2020 with the same period in 2013 to 2019. And while total suspended matter and turbidity typically increases between March and April each year, during 2020 we saw some strong decreases at the most southern stations. The direction of change was mostly similar for the other water quality indicators between 2013 and 2020. We then studied the changes in water quality in the in situ observations. The in situ observations from May 2020 provided us with an opportunity to study water quality later during the lockdown, where remote sensing observations were obstructed by cloud cover. And although seasonal and interannual variations exist, the in situ observations from May 2020 were compared with those collected in May 2019. So we observed lower total suspended matter and turbidity, again in the southern stations in May 2020, but higher values were observed in the northern stations. So chlorophyll A was lower or similar in May 2020 compared to May 2019, while the absorption of color dissolved organic matter was higher at most stations in 2020. And the FU classification of water color was not measured in May 2020, so we were not able to address any changes in this water quality indicator from the in situ measurements. Having observed an improvement in water quality in Lake Vembanad during the lockdown, we were interested whether this observation lasted. We therefore studied the change in water quality indicators after lockdown measures were lifted by comparing the remote sensing observations preceding the lockdown with those from October and November, once cloud cover had lifted. And results showed that the observed improvement in water quality during lockdown was no longer evident. So instead, changes in water quality indicators were more variable in this post-monsoon period, and they were likely related to the influence of rainfall during the monsoon and associated changes in the hydrology of Lake Vembanad. To place the observed changes in water quality in 2020 in the context of longer-term changes, we analyzed interannual trends in the 5 satellite-derived water quality indicators between 2013 and 2020. And this was the full satellite record available at the time. And so results show that all water quality indicators decreased at the northern locations and increased at the southern locations. And at the central locations, opposing trends were observed, with total suspended matter and the absorption of color-dissolved organic matter and turbidity decreasing, and chlorophyll A and the FU classification of water color increasing over time. So this shows that, for example, for total suspended matter and for turbidity, the long-term trends in the southern station showed a different direction of change, which was positive, than we observed during lockdown, which the direction of change was negative. I showed that water quality improved in large areas of Lake Vembanad during April and May 2020. As evidenced by degrees in total suspended matter, absorption by color-dissolved organic matter, and turbidity. A similar change was not observed during the same period in preceding years, suggesting that the reduction in anthropogenic activities associated with the nationwide lockdown in 2020 had a positive effect on water quality. High concentrations of suspended and dissolved matter in Lake Vembanad have previously been related to urban, agricultural, and industrial activities, and restrictions to those anthropogenic activities at specific locations may explain why the observed improvement in water quality was strongest in the central and southern regions of Lake Vembanad. A halt to transport, industry, and hospitality services could have had a positive effect on water quality in the entire region, while the confinement of people to their homes during lockdown may have led to increased domestic waste and sewage discharge in highly populated areas in the north of Lake Vembanad. Other processes, such as reduced atmospheric and wind-induced deposition of fine particles, may have further contributed to an improvement in water quality. Although we were not able to pinpoint specific sources of pollution, our study showed that water quality can improve substantially when anthropogenic activities are reduced. And this provides important insights into the future management of Lake Vembanad and other coastal and aquatic ecosystems. We showed that a coordinated response in reducing anthropogenic activities can improve water quality, with the potential to sustain and restore the ecological values of Lake Vembanad in the future. And continuing such a coordinated response could help achieve the targets set out in the United Nations Global Development Goals and significantly reduce aquatic pollution and improve water quality by 2030. As I've shown, remote sensing observations can aid in this, with free and open data available at high resolutions, noting that it is important that region-specific satellite retrieval algorithms are developed. And the development of such regional satellite retrieval algorithms for Lake Vembanad is what I will be discussing as part of the second case study. As has been mentioned on this training course several times now, the validation of satellite-based observations is essential. In the process, we verify that satellite-derived observations are a true representation of what is happening in the field. There are different approaches to validating satellite observation, and a common approach is the direct comparison of satellite data with in situ observations. in which the in situ data is matched to satellite observations at the same time and the same location. With statistical analysis of the match-up data, we can then determine whether there's a good fit between in situ and satellite data or a poor fit. If there's a poor relationship, we then might search or we might do further research to develop and improve existing algorithms for specific regions of interest. During the first case study, I mentioned that not all of the existing satellite retrieval algorithms that we use to study water quality in Lake Van Bernard performed as expected. And in this case study, we therefore developed regional algorithms for chlorophyll A and total suspended matter. And we follow a slightly different approach than the commonly used method that I just referred to, because if you remember, we had very little in situ data available for match-up. Before I go into detail about how we develop our regional satellite retrieval algorithms for Lake Van Bernard, I wanted to show you an example of a direct validation of satellite data. So in one of our research projects related to the ocean carbon cycle, my colleague Christina Kong validated several satellite-based algorithms for particulate organic carbon. And in her case, a large global data set of in situ measurements of POC was available. And she updated this data set to increase the number of observations to just under 6,000 globally. She then used the data from the Ocean Color Climate Change Initiative, which is a long-term climate quality ocean color data set that I think TJ will be talking about in some of the next lectures. And she used this data set to match up the in situ and satellite data at all of the locations and times of the in situ global data set. As you can see in the middle figure, Christina managed to match up just over 3,000 data points, which you could then use to validate the 8 different satellite algorithms for POC, which is shown in the figure on the right with the in situ data on the x-axis and the satellite-derived data on the y-axis. And so in these results, Christina already reparameterized the algorithms, and you can see that some performed better than others. And in this case, the statistical analysis showed that algorithm B in panel B was the best performing POC algorithm at the global scale. So this is just an example of a really elegant comparison of in situ and satellite data. The example from Christina shows how validation and algorithm tuning can be carried out when thousands of in situ data points are available. But if you remember from the first case study, I mentioned that for Lake Van Bernard, we only have between 8 and 22 match-up data points available, depending on whether we are looking at Landsat 8 or Sentinel-2 data. So what to do if such a limited number of match-up data points are available? My colleague Farinan followed an approach to validation and algorithm development in which modeling techniques were used to obtain the inherent optical properties and the remote sensing reflectances using both local in situ data and in situ data from other sources. And I will talk you through his approach and the development of regional satellite-based algorithms for chlorophyll A and total suspended matter for Lake Van Bernard in the rest of this case study. So in short, in situ measurements from Lake Van Bernard and a database from NASA were used to model absorption and backscattering coefficients to obtain model-derived reflectances. These model-derived reflectances, together with the satellite match-up dataset, were used to correct satellite-derived reflectances, which were obtained from different atmospheric correction methods. And the bias-corrected satellite reflectances were then used as an input to the regional satellite-derived algorithms for chlorophyll A and for total suspended matter. I realize that this diagram is complex, and I will talk you through all of the different steps in more detail, starting with the in situ data sets that Farinan used for this study. So one of the in situ data sets that we used in this study is the one I mentioned earlier for the research on water quality in Lake Vembanot. Just to remind you, for this data set, we carried out field campaigns between May 2018 and May 2019, with samples being collected every 20 days at 13 different locations in Lake Vembanot. As I mentioned before, many different measurements were carried out as part of these field campaigns. And for this study, we used the concentration of chlorophyll A, the absorption of particulate matter, and the concentration of total suspended matter. And this data set contained between 162 and 228, so around 200 data points, depending on the variable. The second in situ data set that we used is the NASA Bio-Optical Marine Algorithm dataset, or NOMAD, which is freely available online, and we used the version 2 dataset for this case study. And from this dataset, we also used the concentration of chlorophyll A and the absorption coefficient of particulate matter, but we also used the absorption of non-algal suspended particles, the absorption of color-dissolved organic matter, the water-leaving irradiance, the downwelling surface irradiance, and the backscattering coefficient. And this data set contains between roughly 100 to 800 data points, depending on the variable. So this adds a lot more in situ data to our analysis than we have from Lake Vembanot alone. So that in situ data that we used in this case study, and I will continue on with the satellite observations that we've used. We used Sentinel-2 data in this case study, for which level 1C products were downloaded from the Copernicus Open Access Hub. And again, just to repeat, this data is freely available and you can download the data through Copernicus. But it's also available from cloud services such as Google Earth Engine Data Catalog. So for the regional algorithm development, we tested different atmospheric correction methods, and we processed the Sentinel-2 data using Acolyte and Polymer. Acolyte is based on a dark spectrum fitting approach, while Polymer removes the atmospheric signal and estimates the water-leaving reflectances using a polynomial function of the wavelength. We also created a match-up data set with the in situ measurements from Lake Vembanot, and we had 12 match-up data points for Sentinel-2, and we had that was processed with Acolyte, and we had 14 match-up data points for Sentinel-2 data that was processed with Polymer. So the next step that was performed is the modeling of the inherent optical properties. So that is the absorption and the backscattering. And we used both the Lake Vembanot database for that and the NOMAD in situ database. I start with the absorption model, so in which total absorption is the sum of the absorption from water, phytoplankton biomass or chlorophyll A, non-algal suspended matter, and color-dissolved organic matter. So to estimate the absorption of these constituents in the water, different models were used that are listed in the table, and you can find the details of those models in the references. The figure on the right shows the comparisons of these biomass, suspended matter, CDOM, and total absorption models from left to right, with the in situ data per wavelength, so wavelength from top to bottom. The in situ data is green for Lake Vembanot and then is gray for the NOMAD database. So in general, these absorption models performed really well. We do see some deviation in the model at higher wavelengths, for example, in the absorption of CDOM, but this did not affect the results of the total absorption model, which is in the most right column, as the contribution of CDOM is relatively small compared to the other constituents in the water. So I continue with the particle backscatter model, in which the total backscatter coefficient is the sum of the backscattering coefficient of water and that of non-algal suspended matter. And in the case of Lake Vembanad, we assumed that the backscatter coefficient of chlorophyll A is small compared to that of non-algal suspended matter, and it's therefore not included in our particle backscatter model. Again, we used models from literature as listed in the table, and more details can be found in the references listed. When it came to the modeling of particulate backscatter, we faced a conundrum. Neither the in situ dataset from Lake Vembanad nor that from the NOMAD database included all the elements that we needed for the particle backscatter model. The in situ dataset from Lake Vembanad included measurements of the concentration of total suspended matter, which is S, and the absorption of non-algal suspended matter, which is AS, but not the backscattering of those particles. The NOMAD dataset included the backscattering and the absorption of non-algal suspended particles, but not the concentration of total suspended matter. We therefore used the common measurement in both the in situ datasets, the non-algal suspended particle absorption coefficient, the AS, to link the backscattering of non-algal suspended particles, BBS, to the concentration of total suspended matter, S. And so we used the model of Balasubramanian et al. And for the NOMAD dataset, the backscatter of non-algal suspended matter was described as a function of absorption, as you can see on the figure on the left. And this modeled backscatter coefficient was then compared with the in situ data from NOMAD as shown in the figure of the right. The results showed that the backscatter model was in good agreement with the in situ backscatter coefficients from the NOMAD dataset at different wavelengths. The modeled inherent optical properties were then used in a forward reflectance model to obtain model-derived reflectance values. And these model-derived reflectance values were then matched up with the reflectance from Sentinel-2 that we obtained after processing of the level 1C data with acolyte and polymer. So the forward reflectance model is a function of the total absorption and the backscatter coefficients as shown in the equation. And this model is applied using the modeled inherent optical properties from the NOMAD in situ data set as well as the in situ data set from Lake Vembanad. And the figure on the right shows the comparisons between the measured and the modeled reflectances from the NOMAD dataset in gray and the modeled reflectances from the in situ dataset from Lake Vembanad and the satellite-derived reflectance routes from acolyte in red and polymer in blue. So the Sentinel-2 data was bias corrected in this process. So the uncorrected data is shown in the lighter blue and red colors, and the bias corrected data is shown in the darker red and blue colors. And overall, you can see that most data points after bias corrections are close to the 1-in-1 line, but there's some variation. What I would like you to notice in this figure is that by using the NOMAD in situ data set, so the bullets or the points in gray, we are able to perform our comparison between modeled and measured reflectances on many more data points than we would have been able to do if we only used the in situ data from Lake Vembanad. So it has huge value to use the NOMAD data set in our case. The modeled reflectances were then used to develop regional satellite algorithms for the retrieval of chlorophyll A and total suspended matter. And the table provides an overview of the tested algorithms. So to estimate chlorophyll A, which is B in this table, the algorithms of O'Reilly based on a blue-green reflectance band ratio and Gillerson et al. Based on the near reflectance band ratio were examined, as those are commonly used algorithms for open ocean waters and for more complex coastal waters. And then two new algorithms to retrieve chlorophyll A were also formulated based on different combinations of reflectance bands and based on a multilinear model. For the estimation of total suspended matter, the algorithms from Miller and McGee and Netschat et al. Were tested, and it was previously reported that the reflectance in the red bands can increase with the concentration of total suspended matter, especially in sediment-dominated turbid water such as Lake Vembanad. And therefore, a new algorithm is also tested, which is based on a polynomial relationship between the reflectance and the concentration of total suspended matter. And so the performance of all of these algorithms was tested using statistical analysis. And the results showed that the existing algorithms for the retrieval of chlorophyll A were not suitable for Lake Vembanad, even after regional tuning of the algorithms. And the Gillerson et al. Algorithm showed better performance with regionally tuned coefficients at higher chlorophyll A concentrations, but high variance remained at low concentrations for this algorithm. So the new locally tuned multilinear regression algorithm for chlorophyll A performed the best in Lake Vembanad in comparison with the other algorithms that we've tested. And for the estimation of total suspended matter, the existing algorithms actually performed relatively well after regional tuning, but the new polynomial algorithm for total suspended matter performed best in Lake Vembanad. So we now have two regional algorithms that we can work with in Lake Vembanad. So as a final step, the best performing regional satellite retrieval algorithms for chlorophyll A and for total suspended matter were validated using the in situ data set from Lake Vembanad. And so the in situ concentrations of chlorophyll A and total suspended matter were compared with the satellite-derived concentrations from Sentinel-2. So again, in red for acolyte and in blue for polymer process data. And we also compared this for the modeled reflectances in gray. And there's a good fit with the modeled reflectances and a reasonable fit with the Sentinel-2 reflectances, with some outliers that originate from one specific date. And so the comparison of the atmospheric correction methods showed that acolyte was the best atmospheric correction method for retrieving chlorophyll A, and polymer was the best atmospheric correction method for retrieving total suspended matter. The figures on the right show an example of the application of the selected regional algorithms and the best performing atmospheric correction scheme for both chlorophyll A and total suspended matter, with examples for two different days in Lake Vembanad. So our study included an extensive field campaign in Lake Vembanad that measured various water quality indicators over an annual cycle, but only a small number of match-up data points were available. Matching satellite and in situ data in Lake Vembanad was difficult for several reasons, including logistical issues and conflicting requirements to affect the scheduling of the field campaigns during satellite overpasses. Carrying out simultaneous in situ measurements at 13 different stations across a 100-kilometer long lake is of course difficult. And the frequent cloud cover, especially during the monsoon season, limited satellite retrievals at those times. We therefore employed a modeling approach to estimate the reflectance values based on inherent optical properties that we either observed or estimated from in situ observations. And the simulated reflectances constituted a large data set of about 160 data points for Lake Vembanad that could be used to improve satellite algorithms in the region. And using this approach, we demonstrate that the locally tuned algorithms are performed better than existing models, which is very promising. And we are now currently working with the new algorithms to continue our research. The atmospheric correction of Sentinel-2 data can be challenging in complex water, and regions with poor air quality, high aerosol content, and high humidity, such as Lake Vembanad, can add to that. We therefore tested two different atmospheric correction methods that are known to perform well in complex situations. The reflectance values retrieved from both acolyte and polymer showed a significant offset when we compared them with our simulated in situ reflectances. And the data needed spectral bias correction to bring the magnitude and spectral shape to within reasonable values. The underlying cause of this remains unknown, but we could speculate that the methods were unable to distinguish fully between high aerosols in the air and scattering particles in the water. We also note that the choice of an atmospheric correction method in our case depends on which variable is being retrieved. And although both atmospheric correction methods produce similar results, we recommend that the use of acolyte for the retrieval of chlorophyll A and polymer for the retrieval of total suspended matter in Lake Vemmenat. I move on to our third case study, which is focused on the mapping of Vibrio cholera bacteria in the environment. Shuba already provided many examples of how satellite data can be used for studying waterborne diseases in relation to human health. And I will focus on our own work in Lake Vemmenat, where we develop a local algorithm to map the risk of Vibrio cholera in the environment using one of the water quality indicators, which is chlorophyll A. As I mentioned early on in my lecture, waterborne diseases are endemic in the Lake Vemmenat region, and both diarrhea and cholera remain important public health problems in Kerala. Public health data shows at least 2 outbreaks per year in the past decade, but we also know from World Health Organization reports that less than 16% of cases are reported in India. And although we understand the direct routes of infection quite well, for example, through the use of contaminated water, there's much to learn about the indirect routes of infection, such as those through the environment. So for example, Shuba talked about the temperature and salinity conditions in aquatic system in which pathogens such as Vibrio cholera can survive. But she also talked about the association of Vibrio bacteria with phytoplankton and zooplankton, and how these variables can be used to model the risk of pathogens in the environment. In the case of Lake Vemmenat, we were interested in whether we could use relationships between pathogens and environmental variables that are amenable to remote sensing to map the risk of Vibrio cholera in the environment. For this case study, we used the same in situ data set from Lake Vemmenat that I've mentioned before, and the data was collected every 20 days between May 2018 and May 2019 at 13 locations in Lake Vemmenat. And in this case, for this case study, we looked at the environmental variables such as temperature, salinity, nutrients. We looked at water quality indicators, including total suspended matter, turbidity, and water color. We looked at chlorophyll A and phytoplankton specific composition. And we looked at human pathogens such as Vibrio cholera and E. coli. And I would like to note here that we've been collecting more in situ data since 2021 to continue our research on waterborne diseases in Lake Vemmenat. And our field campaigns now extend to coastal areas as well. The satellite data that we used in this study is again level 1C data from Sentinel-2, which I obtained from the Copernicus Open Access Hub. And we processed this data using acolyte to perform the atmospheric correction and obtain the remote sensing reflectances. We then used an existing chlorophyll A algorithm because at the time of this study, we did not yet develop our regional algorithms for chlorophyll A. So the first step in mapping disease risk from satellite observations is to analyze the in situ data and studying empirical relationships between pathogens and variables that are amenable to remote sensing. So those variables can include temperature, salinity, chlorophyll A, phytoplankton size classes, total suspended matter, turbidity, water color, the attenuation of light. So there are many potential variables we can look at. And so based on earlier results in other regions, we first studied the relationship between Vibrio cholera and physical chemical conditions in Lake Vembanad, but we could not find clear relationships between, for example, Vibrio bacteria and temperature and salinity. So we then studied the environmental reservoirs of Vibrio cholera in Lake Vembanad, and we see that Vibrio cholera bacteria are almost always present in the lake, and that there is a strong association with phytoplankton. We then studied this association of Vibrio cholera with phytoplankton in more detail by looking at Vibrio bacteria in the environment relative to phytoplankton biomass. And we used the presence and absence of genes of the pathogenic strains of Vibrio cholera in our samples to estimate the probability of those pathogenic strains in the environment for set concentrations of chlorophyll A. And the figure shows the results of this analysis with a positive relationship between Vibrio bacteria and phytoplankton in the north of Lake Vembanad and a negative linear relationship in the south of the lake. I would also like to point out in this figure that the risk of Vibrio bacteria in the environment is actually always high, with probabilities between 50 and 100%. So the empirical relationships between Vibrio cholera bacteria in the environment and chlorophyll A that we obtained from the in situ observations were then used in combination with satellite-derived observations of chlorophyll A to map the risk of Vibrio cholera using Earth observation, which you can see an example of in this figure. So you see higher risk in the northern part of Lake Vembanad, where there is a positive relationship, and lower risk, but still high, in the southern part of the lake, where there's a negative relationship between Vibrio cholera and chlorophyll A. So in this case study, I've shown you that in situ data can be used for the development of novel satellite algorithms. And in this case, we used the relationship between Vibrio cholera in the environment and chlorophyll A that is amenable to remote sensing to map the risk of pathogenic bacteria in Lake Vembanad using Sentinel-2 data. And I showed that we observed different relationships in the north and the south of Lake Vembanad, which might partly be explained by the differences in the environmental conditions between these two regions of the lake. There's a man-made barrier in the lake, which causes more freshwater conditions in the south of the lake and more saline conditions in the north of the lake. And these differences in conditions are likely to support different phytoplankton communities. As Shuba mentioned last week, it is known from literature that some phytoplankton species attract Vibrio bacteria, whereas others deter the bacteria. So our research is ongoing in trying to understand this better and the ecology of Vibrio cholera and other pathogens in Lake Vembanad. So I will now continue with the fourth and last case study on flood mapping, in which we use both multispectral observations from Sentinel-2 and synthetic aperture radar observations from Sentinel-1, and validate the satellite observations using local information. If we look at the total disaster events by type since the 1980s, in the figure at the bottom left, we see that floods are the most common natural disasters worldwide, with over 3,000 events in the last 20 years. We also see that the occurrence of floods worldwide has increased considerably in the past decades, with over a doubling of events from the period of 1980 to 1999 to 2000 and 2019. Floods are also the most common natural disaster in India, with reports of nearly 1,300 deaths and an economic cost of $3.1 billion in just 2021. Floods can have major consequences, with the potential to destroy life, displace populations, break down infrastructure, destroy sanitation facilities, and impede access to food, clean water, and medicine. Because of this, floods are often associated with outbreaks of waterborne diseases such as cholera. Again, for this topic, it's important to consider the new United Nations Sustainable Development Goals, including SDG 3 for good health and well-being, 6 for clean water and sanitation, and 14 for life below water. And if we look at some of the targets of Goal 3, we see that one of them aims to strengthen the capacity of all countries, in particular developing countries, for early warning, risk reduction, and a management of national and global health risks. Strategies for attaining those targets must include improved responses to flooding, and a first step in the response must be the identification of flooded areas. The mapping must cover the entirety of the affected area, and it has to be rapid to be effective. And in this case study, we present an application of remote sensing to map flooded areas using the Femelot coal wetland system as an example. When we started our project on the environmental reservoirs of waterborne diseases in Lake Femelot in 2018, the southwest of India experienced an extremely heavy monsoon season. And as you can see in the graph in the middle, rainfall was considerably higher in 2018 compared to the average rainfall in the previous decade. So this extremely heavy monsoon season in 2018 caused once-in-a-lifetime floods in the Lake Femelot region in August, with many people being displaced and over 300 lives lost. This was reported in news outlets worldwide, including the BBC in the UK. And quite quickly, NASA shared an RGB image of the before and after of the floods, which were based on Landsat data. And I was working with multispectral satellite images at the time, so I posed the question whether multispectral images can be used to delineate flooded areas. To map floods, we followed a quite straightforward approach based on freely available satellite images, software, and existing satellite retrieval algorithms. I downloaded Sentinel-2 images, again from the Copernicus Open Access Hub, and we processed the data using ecology to perform the atmospheric correction and obtain the remote sensing reflectances. We then used existing algorithms to distinguish water from land, which I will explain a bit more later in the next slide. And then used the threshold value to generate binary maps of water and land, and used several clear sky images to generate a base map of Lake Femelot that covers the normal extent of the water in the lake and the surrounded areas. So the water extent when there's no flooding. The flood maps were generated by determining the difference between the base map and the binary water index map for a specific day. A similar process was followed for the Sentinel-1 synthetic aperture SAR data, which is regularly used for flood mapping applications. So again, the data was downloaded from Copernicus, and in this case, I processed the data using the ESA SNAP application to obtain the backscatter. The backscatter was then used as an index of water, and we follow a similar approach to the S2 flood mapping to generate a base map and flood maps. For the S1 data, we followed the method described in the United Nations Spider portal, which I think has been mentioned before, maybe during this training course. It provides a step-to-step guide, and it explains how to download the data and how to generate flood maps using SNAP. And so I provide a link to this at the end of my presentation. I think it's a really useful resource, and you can learn some cool techniques just following those instructions. So following those methods, we have studied the accuracy of the S2 flood maps in comparison to the S1 maps using 4 different water indices, including the normalized different water index, the modified normalized different water index, the water ratio index, and the automated water extraction index. And these satellite retrieval algorithms are used to distinguish water from land pixels using different sets of Sentinel-2 bands including the green, red, the near, the mere, and the swear bands. All of these algorithms have been published, and you can look up the equations in the references from the table. So just to show you an example of the different algorithms after processing of the data, here are the maps of the 4 water indices with from left to right, the NDWI, the modified NDWI, the AWEI, and the WRI. So we tested the applications of these algorithms for flood mapping using multispectral remote sensing data, after which we selected the best performing algorithms to address the mapping of the once-in-a-lifetime floods in August 2018. And during flood events in 2021, we then validated the S2 flood maps in Lake Vembanot region using information on paddy field cultivation cycles in the south of Lake Vembanot. So these are the results of the Sentinel-2 flood maps for 4 different water indices, with from the left to the right an RGB image from the 29th of January 2018, a map of the water index, the associated histogram with threshold indicated, the map of the binary water index, the base map of that specific water index, and then the flood map with in red the pixels that are indicated as flooded. So those are the pixels that are outside the regular extent of the lake. What we can see is that the WRI shows the least detail, and the other water indices show much more detail with flood pixels indicated mostly towards the southwest of Lake Vembanot. And we see a similar result in the SAR observations. So those are from the same day, the 29th of January in 2018. And then we compared the Sentinel-2 base maps and flood maps with those of Sentinel-1, using a confusion matrix to determine the overall accuracy of the Sentinel-2 flood maps. And so when we do this comparison between the S1 and the S2 data, we see that the base map and the flood map of the modified normalized different water index has the highest overall accuracy. We then use these M and the WI to map floods in August 2018, as you can see here on the left. And the main area of flooding was towards the south of Lake Vembanot. And when we zoom into this region, we can see that the flooded area on the RGB image having a slightly more yellow color than the surrounding land. And that's also indicated in red in the flood map in the center. We then compared the Sentinel-2 flood map from the 21st of August with the Sentinel-1 SAR image of a day later. We didn't have matching days, but they were close enough to compare. And we showed that the accuracy of the Sentinel-2 map is high, with pixels in red indicated as flooding in both the Sentinel-1 and the Sentinel-2 data, pixels in orange indicated as flooding in Sentinel-1, and pixels in dark blue indicated as flooding in the Sentinel-2 data only. And so the overall accuracy of the Sentinel-2 based map was 88%, and in this example of the August 2018 floods. Since 2018, the Lake Vembanot region has experienced more extreme flooding events, also in October last year. And we took this opportunity to validate flood maps by scanning local newspapers for reports of flooding. And we could confirm that regions in the south were indeed flooded areas. But the regions closer to the lake were paddy fields used for cultivation of rice. So we know that those areas might have not been flooded. So what we did is we selected 16 different paddy fields and identified the months and years where these paddy fields are expected to be flooded according to the cultivation calendar. And so we prepared masks for these paddy fields, so we can exclude them from our analysis during those specific months, which allows us to more accurately assess the extent of flooded areas throughout the year. So to summarize and discuss, I have shown that we can indeed use multispectral remote sensing images from Sentinel-2 for flood mapping in the Lake Vembanot region. Although there are also limitations in the application of the Sentinel-2 data because of cloud cover, of which an example is given here. We see that if there is thick cloud cover, we are not able to map the M and the WI or the flooded region correctly. Nevertheless, we consider flood mapping using multispectral remote sensing data a valuable addition to the existing technique based on the SAR data, with Sentinel-2 images available every 5 days for Lake Vembanot and Sentinel-1 images only available every, I think it's now 20 days. So the use of Sentinel-2 data increases the temporal resolution considerably. Another benefit of the Sentinel-2 data is that we can simultaneously map water quality, as I've shown in the first case study, and also map waterborne diseases, as I've shown in the third case study, and both can provide important additional information in response to natural disasters. If you are interested in finding out more about our work in Lake Vembanot, our team has published a couple of papers related to water quality and waterborne diseases in a special issue in remote sensing, which was focused on Earth observations and sustainable development in marine and freshwater systems. The work on water quality has been published in this special issue, so if you want to find out more about the methods used or the results, then please have a look at the special issue. And I think the work from Anas on the cholera algorithms and the observations of cholera in Lake Vembanot has also been published in that special issue. And as I mentioned before, if you want to do some flood mapping in your own region, I would advise you to start with Sentinel-1 method and follow the step-by-step guides on the UN Spider portal, which is a really good resource. And then, as I mentioned at the start of my lecture, the work I presented today has been carried out by a large team of scientists and students, and I would hereby like to thank all of them for their contributions. Some of them are in the panel, so hopefully they can help me with answering some questions. And that was it. Thank you for listening, and please feel free to ask questions in the chat if you haven't done so already.